morning to all of you. I know it is very difficult to sit in this humid morning because of the rains. Of course, shortly the rains may intensify because the monsoon has reached. So now we have another 3-4 months of heavy rainfall in this region. Along with that, the rains, we will also continue our process of teaching and learning. In the topic that we have decided discussing, that is the cell and its organelles and their function. Last class, just I have introduced about the mitochondria to all of you. Of course, you know many things about mitochondria because you have been studying this cell from your high school or even primary school level. So that's why you have some basic idea about all the cell organelles. So that's why my responsibility today is to first rebrush what you have learned over these years and add few more things to your to your information or for your understanding. Okay, you know the mitochondria is the powerhouse of cell, and why it's called as powerhouse of cell also you know because it undertakes the aerobic respiration where with the use of oxygen it is able to form more and more ATP molecules which are considered as the energy currency of the cell. So this mitochondria, they are almost rod-shaped organelles, capsule-like thing. I might have told you in the last class, 3 millimicrons to 5 millimicrons in length and 1.5 to 2 millimicrons in diameter. This was first observed by Richard Altman, and the term mitochondria was coined by Carl Benda in 1898. The literary meaning is mito means thread, chondrion means granule-like. Granule-like threads, that's the literary meaning of the cytoplasm. Of the mitochondria which is found in the cytoplasm. And this mitochondria contains two membranes, outer membrane and inner membrane. There are two membranes. But these two membranes are not firmly sandwiched. Instead of that, there is a space between inner matrix and inner membrane and outer membrane also. That is known as perimitochondrial space. I will come to that later. Inside the inner membrane, there is a large space. That is known as the matrix of the mitochondria. In this matrix, you can see which is moderately dense, and you can see the strands of DNA, ribosome, and small granules. The very interesting fact here is the presence of DNA, because DNA being the nucleic acid, we always think it is inside the nucleus. But for the fact that the mitochondria have their own DNA, which is mostly circular DNA. Mitochondria is generally called as the powerhouse of cell. Why? The question is, of course, all organisms need food. We eat food or the plants prepare their own food. Why the food is necessary? In order to produce high energy electron. And this high energy electron is used to store the energy. That means especially the energy is stored in the phosphate bonds, energy phosphate bonds in a molecule, and this energy phosphate bonds which store the energy, we call it as adenosine triphosphate or ATP. So, whatever the food that we eat should be ultimately able to produce the ATP molecules, which are called as the energy currency of the cell. And the energy here is stored in the high energy phosphate bonds. This ATP is converted from ADP, adenosine diphosphate. If you add one phosphate group to ADP, that is adenosine diphosphate, it becomes adenosine triphosphate. Uh, so various reactions in the cell, they use the energy. Where what they do is they use the energy stored in this high phosphate bond and convert the ATP into ADP. And ADP becomes energy rich by taking one more phosphate group, and then it becomes ATP. So that's why this is a continuous process. So now uh, now this conversion of ADP to ATP, adenosine diphosphate to adenosine triphosphate, it is done in the, especially in this mitochondria. So these mitochondria are responsible for the conversion of nutrients into energy yielding molecule. Whatever the food that we eat, ultimately that is being converted into adenosine triphosphate, and that is the fuel for all cellular activities. That is the fuel for all cellular activities. And this function of preparing this fuel is called as aerobic respiration. And this mitochondria is the site of aerobic respiration, where the energy is produced and stored. So that's why the mitochondria is known as the powerhouse of cell. So this aerobic respiration involves three important: glycolysis, Krebs cycle, and ETS, electron transport system. With the help of these three biochemical reactions, the mitochondria is able to produce the energy. Now, from where these mitochondria are derived? Why it is important to see? See, when the first cell was formed, very primitive cell was formed, there was no mitochondria in it. Because all of you know, this first cell or protocell or very primitive cells, they were, or even living beings were anaerobic, that is, used to undertake all the activities in the absence of oxygen. So probably the mitochondrial origin is coupled with the evolution of oxygen on the earth. When the oxygen increased in the atmosphere, probably the cells must have thought of gaining a new organelle which can make use of this oxygen, or which can utilize this oxygen. So by Dino, we don't know how exactly, but there are certain theories. One is the mitochondria is derived from a bacteria. The mitochondria derived from bacteria by a process what is known as endosymbiosis. Endosymbiosis. What is symbiosis? If two organisms or two things are interrelated depending on each other, we call it as symbiosis. This is endosymbiosis, where one is inside the other and both of them mutually get benefited. So mitochondria must have arose about 2 billion years ago, when a bacteria, when a bacterium fused with a primitive cell, that is the archaeal cell, or This bacteria and the archaeal cell must have developed some symbiotic relationship with a primitive eukaryotic cell, very primitive eukaryotic cell. Then, as a result of this, the mitochondria must have been formed. Because the closest extant relative of bacteria which give rise to mitochondria are rickettsia. The bacteria that give rise to mitochondria are rickettsia bacteria. And the first person to recognize the mitochondria as a descendant from endosymbiotic bacteria was Evan. So here, endosymbiotic theory can be understood in three steps. First, see, this is a proto-eukaryotic cell, very primitive cell, archaic cell. Infolding of the plasma membrane of the ancestral prokaryote gave rise to endomembrane components, including nucleus, endoplasmic reticulum and everything. So what has happened from this one, the infolding of membrane, the membrane started infolding and that gave rise to many of the cell organelles. In the first endosymbiotic event, probably the archaic cell or ancestral eukaryotic cell consumed a bacteria, must have consumed a bacteria, aerobic bacteria, a bacteria which can make use of the oxygen. That must have evolved into what you call the mitochondria. See, aerobic bacterium, it was got fused, and then this bacteria was an aerobic bacteria which has the capacity to utilize the oxygen. Then this must be a photosynthetic bacteria also, and this aerobic bacterium developed into what you call the mitochondria. This is what endosymbiotic theory suggests. So that's why if you talk about the origin of this mitochondria, the cell along with the mitochondria grew by importing most of their protein from the cytoplasm and also by internal synthesis of some protein and replication of the gene. The cell, of course, harbored mitochondria along with other cell organelles also. That means the cell could grow at this time by importing most of the protein from the cytoplasm only, by internal synthesis. By internal synthesis, some proteins and replication of the genome. There was only this thing. Similar to the cells, even the mitochondria, newly formed mitochondria, started divide and fuse with other mitochondria, thereby maintaining the number of cells. Both fusion and fission was a very common practice there. So, in the process of evolution, this aerobic bacteria ultimately evolved or metamorphosed or changed into a new structure, what is known as this mitochondria. This is how the mitochondria appears. As I have told you, it looks like a capsule. There is two membrane, this is the outer membrane and this is the inner membrane. The space between inner membrane and outer membrane is mitochondrial space, and this is the space what you call matrix. About this I will come to that later. So this, if you look into the morphology, mitochondrial size is 0.5 to 1 millimicron in diameter, and most of the time, but only in fibroblast cells it is elongated and thread-like. Then the number of mitochondria varies and it depends on the size and functional status of the cell. If the cell is more active, it requires more energy, so more number of mitochondria. So the mitochondrial number is not constant, it varies. For example, an average liver cell contains 1500 mitochondria. Uh, One thing I can say that they are more in number in the cells with high energy requirements. For example, sperm cell, muscles, or in flagella, you can see more number of mitochondria. In others, the number will be less. Bean shaped or fibroblast, thread like. This is what I. Now, the very interesting fact about the mitochondria is always maternal in origin. In the sense, whatever the mitochondria that we have received, all the mitochondria that has come from the mother only. Father or the paternal contribution is not there as far as the mitochondria is concerned. Of course, oocyte mitochondria, that is the mitochondria that is found in the egg, are the only mitochondria that is inherited. Even spermatozoa also contain mitochondria, which is the contribution from the paternal side. That also enters into oocyte during fertilization, but gets destroyed during the early embryonic development. Why the mitochondria that has been contributed from the paternal side is being destroyed, till now it is not very clear. So one thing is sure, whatever may be the number of mitochondria that we have, all those mitochondria are the contribution from the maternal side. There is no paternal contribution as far as the mitochondria is concerned. Then let's move on to the structure of the mitochondria. Here you can see 5 parts in the mitochondria: outer membrane, inner membrane, intermembrane space, cristae and matrix. See here you can see this clearly. This is the outer membrane and this is the inner membrane. And the space is known as intermembrane space or perimitochondrial space, this space. And the inner membrane is thrown up into number of folds. Can you make a finger like projections of folds? The inner membrane. And these folds are known as cristae. And inside, the inner membrane encloses some inside space, this space, and that is known as mitochondrial matrix. See, this is the matrix. In the matrix, you can see circular DNA, even ribosomes. Or here, the outer membrane, if you look into the simple phospholipid bilayer, this is nothing but a simple phospholipid bilayer, outer membrane, and it encloses the mitochondria. Entire mitochondria is enclosed by this. On the surface, it has some protein structures. Actually, they are openings, and that is called as porins. And ions, nutrient molecules, ATP, ADP, everything can pass through the outer membrane with ease through these porins, which are nothing but protein channels. Because the mitochondria requires these things in order to undertake the cellular respiration, ion, nutrients, ATP, ADP, which should come from the cytoplasm. How they can come inside the matrix? That is through these protein openings, what is known as porins. Then the second one is the inner membrane. Even this inner membrane is also freely permeable to oxygen, carbon dioxide and water. This inner membrane contains some complex of electron transport complex, the ATP synthase complex and transport proteins. So the inner membrane contains even ATP synthase complex because ATP is to be synthesized and transport proteins. More importantly, there's a very sophisticated ion transport system also exists here. Even some antipode systems. What is this antipode system? They will allow anything inside in exchange with something. Or they will send anything outside in exchange with something. So, they are allowing the exchange of ions between cytoplasm and mitochondrial matrix. This inner membrane is not having any cholesterol or but it is rich in only phospholipid and cardiolipids. See, this is the outer membrane. This is the inner membrane. All membranes have phospholipid bilayer, you can see. The space between inner and outer membrane is known as intermembrane space or intermembrane space. So this intermembrane space is about 70 angstrom unit. This intermembrane space has high proton concentration. Then the cristae. I have told you the cristae are nothing but the folds of inner mitochondrial membrane. So see, these foldings of these finger-like foldings, projections that it makes. Inner mitochondrial membrane, we call it as cristae. And cristae is covered with this inner membrane spheres, and those inner membrane spheres are called as stalked particles or inner membrane spheres, or they look like knobs or heads. Probably in earlier photographs, see, these are knobs like things or head like things which you can see on the inner membrane. They are called as stalked particles or knobs or heads. And these heads, they contain F1 portion and F0 portion. They contain two regions. One is F1 portion and F0 portion for ATP synthesis and oxidation. This intermembrane space is also known as perimitochondrial space. And okay, this is what we have already discussed. Then if you enter into the last part of the mitochondria, that is the matrix, which has been enclosed by the inner membrane. The mitochondrial matrix is having gel-like consistency. Gel-like. And it is dense and homogeneous. Almost 2/3 of total protein of the mitochondria that is found in the mitochondrial matrix. 2/3 of total protein that is found in mitochondria that is in this matrix. In addition to this, the matrix also has enzymes, ribosomes, DNA, messenger RNA, granules, fibrils and tubules. The major enzymes are the enzymes which are involved in the synthesis of nucleic acid and proteins, even the enzymes which are required for um, fatty acid oxidation, or the enzymes that is required for TCA cycle. Except succinate dehydrogenase, all other enzymes are formed. After studying the structure, we should also know why these mitochondria are very important. All of you know we eat food, and that the food that we eat, it has basic chemicals, the cells which should be able to make use of that. And some of the best energy supplying foods, they contain sugar or carbohydrates. Now, the sugar are to be broken down by the enzymes, so that the sugar is broken down or split them into the simplest form of sugar, what we call it as monomer or glucose. Then only the glucose enters the cell by special molecule called the glucose transporter. There's a special molecule in the membrane, that what is known as glucose transporter, which can pump the glucose inside. Once the glucose reaches the cell, glucose is broken down into make ATP, adenosine triphosphate. Glucose, the most fundamental or basic carbohydrate, is being subjected to the process of breaking down to make the ATP. This breaking of glucose can happen in two pathways or two methods. The first pathway doesn't require any oxygen. It doesn't require any oxygen, and that is called as anaerobic metabolism. Probably if you recollect, glycolysis, which occurs in the cytoplasm outside the mitochondria, it's an anaerobic breakdown in the absence of the oxygen, and that happens in the cytoplasm or outside the mitochondria. So during this glycolysis, the glucose is broken down into pyruvate. Now, in each reaction is designed in such a way that it should be able to produce some hydrogen ions or electrons that can be used to make energy packets or ATP. Here only 4 ATP molecules can be made by one molecule of glucose run through this pathway. Only 4 molecules. That is why here the mitochondria and oxygen are very, very important. Why? In the first step, that is in the glycolysis, glucose is broken down into pyruvate. Now, we need to continue the breakdown process. What we will make? We will subject this pyruvate in the Krebs cycle inside the mitochondria in order to produce enough ATP molecules to run all the cell functions. First, in the glycolysis, one molecule gives only 4 ATP molecules, but it is not sufficient for us. Still the pyruvate contains the energy. So that's why the pyruvate and is made to enter into Krebs cycle. So this mitochondria are very, very unique in several regards. The one thing is they have their own DNA, that is the circular DNA. They have their own ribosomes, that is the mitochondrial ribosome, the mitochondrial DNA. Okay, this is already discussed. Mother gives genes and cytoplasm to their children in their egg cell. So, since mitochondria are in the cytoplasm and reproduce themselves, they only are inherited from mothers. Now, mitochondria, I have told you that enters into Krebs cycle. Pyruvate, what enters? The pyruvate. Pyruvate is the product of glycolysis. And now, this pyruvate is transported into the matrix of mitochondria. Once it is transported to the mitochondrial matrix, there the same pyruvate is converted into acetyl coenzyme A. Fatty acids from the breakdown of lipids are also transported into the matrix and converted into acetyl co. Not only this glucose, even fatty acids are also pushed to the matrix where they are converted to acetyl coenzyme A. The Krebs cycle then converts this acetyl coenzyme A into carbon dioxide and high energy electron. It is the role of Krebs cycle to convert the acetyl coenzyme A into carbon dioxide and high energy electron. And these high energy electrons are carried by NADH to NADPH, DH2. The next one is electron transport. Still the same molecule can give you energy. So the electron transport, high energy electrons are moved from ADH and FADH2, and this has been passed through three protein complexes that are embedded in the inner membrane. So the final protein complex gives the electrons to oxygen and then converting it into water. As a result, during this process, hydrogen ions come back into the matrix down the concentration gradient, and ATP synthetase, also known as ATPase, the enzyme, uses to generate energy for the formation of ATP from ADP, adenosine diphosphate, and inorganic other phosphates. Now, coming to the functions of mitochondria. Undoubtedly, there is the powerhouse of cell. That's also the center of aerial respiration. There is the respiration with oxygen. And the mitochondria generates metabolic energy, especially 36 molecules of ATP are formed from every molecule of glucose. Although primary function of any of the mitochondria is to convert organic material into biological energy or into cellular energy, which is always in the form of ATP. Mitochondria plays an important role in many of the metabolic tasks that is conducted within the cell. One is, what are those roles? Apoptosis, programmed cell death. It delays actually mitochondria, delays apoptosis. Suppose uh, if there is an injury, it can be rectified. Then the glutamate mediated exotoxic neural injury. It also useful, mitochondria is cellular proliferation, spreading of the cells. Then regulation of cellular redox state, not the oxidized state, but the redox state. Even the synthesis of heme, the iron moiety which is found in the hemoglobin. Then the same thing can produce the steroid synthesis or participate in the steroid synthesis. Then the heat production, especially this is more important among the homeotherms or endotherms, where animals have a constant body temperature. So the mitochondria itself has to produce some heat, as a result of which the organism also stays safe and warm. Then there are certain, these are some general functions, but there are certain specialized functions also. Some of the mitochondrial functions are performed only in specific type of cells, not all. For example, the mitochondria in the liver cell that contain enzymes that allow them to detoxify ammonia, especially in the liver cells, detoxification process takes place. For the detoxification of ammonia, this mitochondria is essential. Ammonia is formed as a result of protein metabolism. This is what I wanted to discuss with you as far as the mitochondria is concerned. I have not got into the details of glycolysis, Krebs cycle, electron transport system, because that will be dealt separately in your physiology class. So that's why, without wasting time, I will move on to the next one, what is known as organelle ribosomes. Actually, I wanted to start with the lysosomes, but let me have the ribosomes first. Another very interesting organelle which is found in the cytoplasm, either freely in the cytoplasm or on the surface of the endoplasmic reticulum. You can see this ribosomes, which are considered as the site of protein synthesis. And these ribosomes are produced in the nucleolar region of the nucleus, in the nucleolus of the nucleus. So this is how the ribosome appear, and ribosomes These are tiny granular structure. Chemically, if you analyze the chemistry, the ribosomes is nothing but are nothing but ribonucleoprotein particle. Ribonucleoprotein particle. Ribosomes serve as a workbench with messenger RNA, ribosomal with messenger RNA acting as the blueprint in the process of protein synthesis. Actually, blueprint for the protein synthesis is brought by messenger RNA. Ribosomes are the site or workbenches on which the proteins are synthesized. The ribosomes were first discovered by studied by Pallade in 1955. Then coming to the number. The number of ribosomes differs greatly. For example, the rapidly growing E. coli cell may have 15,000 to 20,000 ribosomes. Almost 15% of the cell mass in E. coli is because of the ribosomes. Basically, there are two types. One is matrix ribosomes. These are the proteins which remain within the cell, cytoplasm, and another one is the plasma membrane ribosome, which make the protein for transport to outside. These ribosomes prepare certain proteins which act as carrier proteins or transport proteins which lie within the plasma membrane. For a ribosome, there are two domains. One is the translation domain and another one is the exit domain. The reason which is responsible for the translation, okay, of course, if you can recall what you have learnt in the protein synthesis, transcription, translation. So, the reason that is responsible for translation is called as translational domain. A ribosome basically made up of two subunits. They contribute to this domain located in the upper half of the small subunit and in associated areas of the large subunit. This is where the translation takes place. Whereas the other one is the exit domain. Protein, what is the basic structure of the protein? Fundamentals of the protein are nothing but the amino acids. Amino acids which are linked with the help of polypeptide linkage and forms a long polypeptide chain, and that becomes protein later. The growing peptide chain emerges from the large subunit at the exit domain. So, any ribosome has two domains. One is translation domain, where the translation of the messenger RNA coding takes place. Then afterwards a polypeptide chain comes out of that peptide chain. That is what is known as exit domain. Now, ribosomes are not bounded by membranes. Though all other cellular organelles are bounded by membrane, but not the ribosomes. Prokaryotic ribosomes are smaller and less dense than eukaryotic ribosomes. Any ribosome you take, it consists of two subunits. And each subunit is made up of a protein and the type of the RNA, ribosomal RNA. See, this is one ribosome which is made up of two subunits. Each subunit constructed from one to two ribosomal RNA molecules and many proteins. For example, 30S smaller subunit, 50S larger subunit. In eukaryotes, they have 40S, 60S and two subunits, and the ribosome is known as 80S subunit. See, don't get confused. A ribosome is made up of two subunits. In eukaryotes, there are two subunits. One is 40S, S stands for Swedberg unit, 40S, and smaller subunit, and the larger subunit is 60S subunit. And the entire ribosome, which is made out of 40S and 60S, it is called as 80S subunit in eukaryotes. Whereas in prokaryotes, the ribosome consists of two subunits, that is smaller 30S subunit and the larger 50S subunit. And the ribosome is known as 70S type. Now I know many of you have confusion. When there are two units, one unit of 40S and another unit of 60S in eukaryote, how it can be 80S? It should be 100S. See, 40S and 60S, these are the Swedberg unit. That is the sedimentation coefficient. Suppose if you separate the two subunits, larger and smaller subunit in eukaryotes, and put it for centrifugation, then the larger unit will settle at 60 Swedberg and the smaller unit will settle at 40 Swedberg when they are put separate. Suppose if you put the entire ribosome, both the units together, then the entire ribosome with both the units together will settle at 80 Swedberg units. So that's why these two are not additive. When they are separate, this settles at 40 Swedberg, this larger unit settles at 60 Swedberg. When they are together, they settle at 80 Swedberg. Same is true in case of the prokaryotes. One unit is, smaller unit is 30 Swedberg unit and larger one is 50 Swedberg unit when they are centrifuged separately. But when both of them are intact, then the prokaryotic ribosome settle at 70S Swedberg. So that's why it is called 70S type, and eukaryotes it is called as 80S type. There are certain ribosomes which are present in the mitochondrial matrix. Even in the stroma of chloroplast also you can see the ribosomes. Though the mitochondria is in the eukaryotic cell, but the ribosome that is present in the mitochondrial matrix is prokaryotic in the cell. The cell being a pro-eukaryotic, having a mitochondria, should have 80S ribosome. Of course, all the ribosomes in the cytoplasm is 80S. But whereas the ribosomes which are found in the inside the mitochondria or chloroplast, they are 70S. So that's why it is called as prokaryotic type. See, 30S subunit, this is the 60S subunit, 50S subunit. See, when they are together, see. So if you separate them, this will set as 80S, and it will at entire together they will be at 70S, and when they are separated, one will settle at 60s and another will settle as 40s, like that. This is how the 70s ribosome appears. This is the 50s subunit, this is the 30s subunit. Same thing. See, ribosomes, when both the units are held together. Okay. Now, as far as the functions are concerned, I think we can continue in the next class. So, do you have any doubt? ಗೊತ್ತಾಯ್ತಾ? ಕೇಳ್ತಾ ಸರಿಯಾಗಿ? ಹಲೋ, ಕೇಳಿಸ್ತಾ ಇತ್ತು, ಕಾಣಿಸ್ತಾನೂ ಇತ್ತು. ಓಕೆ, ಸೊ ವಿ ವಿಲ್ ಮೀಟ್ ಇನ್ ದ ನೆಕ್ಸ್ಟ್ ಕ್ಲಾಸ್ ಅಲ್ವಾ? ಹಲೋ, ಓಕೆನಾ? ಆರ್ ಎನಿಥಿಂಗ್ ಯು ವಾಂಟ್ ಟು ಡಿಸ್ಕಸ್? ಓಕೆ, ಥ್ಯಾಂಕ್ ಯು ಫಾರ್ ಅಟೆಂಡಿಂಗ್.